All right. All right, so yeah, welcome to the first CHAMP virtual training for 2024. Thank you all so much for joining this program, especially those of you who were able to jump on really in the past week as we've been rolling out the program, trying to get things started before the end of the field season this year. I really appreciate all of you taking part in this active role of stewardship on Lake Champlain. My name is Eileen, and I'm the Education and Outreach Associate with the Lake Champlain Committee. And I'm being joined today by Meg Modley, who's the Healthy Ecosystems and AIS Management Coordinator with the Lake Champlain Basin Program. Just a couple of things before we get started. I mentioned this, we're recording the presentation, so just make sure that you're muted and that your video's off during the duration. You can turn things back on during the Q&A. You can also drop questions as we go along in the chat, and we can open up the chat during the Q&A to get to all those questions at the end. All right. So in this presentation, we're going to cover what aquatic invasive species actually are, why they're an issue on Lake Champlain, actions that you can take to prevent the introduction and spread of AIS, and then we're going to move to what this program is all about and get into the nitty-gritty of what we are monitoring for on the lake, going through each species and ways to identify those species. And then I'm going to take you through how to conduct and submit a survey for those aquatic invasive species. So I'll pass it off to Meg now. She's going to go over all the AIS information.
Hello, everyone. Thanks for having me, Eileen. It's really nice to be here from the Lake Champlain Basin Program, and we've been partners with the Lake Champlain Committee for decades. So it's really nice to share this space. So yes, my name is Meg Modley. I do focus on aquatic invasive species management. I've been with the Basin Program since 2003. And I started working on invasives in particular in around 2006. So starting off with what are aquatic invasive species, and we do have a federal definition that we follow at the Lake Champlain Basin Program, identifying that invasive species are plants, animals, or pathogens that are non-native to the region, meaning not here pre-European settlement, and that have been measured to cause some type of harm. And that's a differentiation between what a non-native species is not here pre-European settlement, and invasive has that added criteria of being measured to cause some type of harm. That harm could be to the environment, to the economy, or to human health. And these invasive species are so prolific because they often can produce self-sustaining populations. And three of the heavy hitters in Lake Champlain currently are on the lower left, dense beds of Eurasian water milfoil. We do have both spiny and fishhook water fleas, which are crustaceans in the plankton community. And we have zebra mussels growing on top of each other in the lower right photograph. Next slide. Thanks. So to sort of figure out or start to wrap our hands around invasive species, folks often wonder how it is that they arrive in Lake Champlain or how they move across the landscape. And often these organisms are moved across oceans or across watersheds with assistance. So one of our big connectors in Lake Champlain to water bodies north and south of us are canalways. We have canals in the Richelieu River, the Chambly Canal, and we have the Champlain Canal to our south, and the Hudson River that connect our watersheds, and species can swim or float or be entrained through those systems. And currently we're working to see if we can find some mitigation measures to help prevent species from moving through the canals. Another common pathway is water garden escapes. We have a lot of ornamental and pretty plants that we like to plant in our water gardens, and we are having more frequent flooding events and tropical storm remnants that are coming through and flooding the areas where species that are either stocked in ponds or in water gardens escape and start to grow in receiving water bodies in the watershed. We have overland transport on boats and trailers. Oftentimes, aquatic plants and animals are growing in and around boat launches and can be transported on anchor lines, in bilge water, in live wells, on the exterior of watercraft, wrapped around boats, hulls, on the trailers, and on other recreational equipment. We have our boat launch steward inspection and decontamination program to address that pathway. And then we also have what I call the free Nemo phenomenon, which is the aquarium dumping. We do have evidence that folks, once their pets outgrow their space in their aquaria or their cages, and they can't take care of them anymore, they decide to release them into the wild. So that can start new infestations growing. And we do have illegal bait introductions or accidental bait introductions if the bait fish regulations are not followed closely in Vermont and New York and Quebec. Next slide. So the current status of known non-native and invasive species in Lake Champlain, we're looking at the lower left graphic, shows over time when we believe these non-native and invasive species were introduced into the lake. So the last introduction was in 2018, a fish hook water flea. We have not documented a new invasive species introduction since then, but I would like you to pay attention to the fact that the darker green or teal color represents those species that we have measured to cause some kind of harm. So that's just about a dozen or so of the 51 known non-native species have been measured to cause some type of harm. Unsurprisingly, we see water chestnut, Eurasian water milfoil, zebra mussels, alewife, spiny water flea in that category. But also like to point out that we as humans have introduced non-native species for our enjoyment in the lake, such as rainbow trout or brown trout. And then the graphic on the right documents the number of known non-native invasive species in the surrounding water bodies that we are connected to Lake Champlain. So while we have 51, we know of closer to 80, 87 in the St. Lawrence, 188 in the Great Lakes, and over 122 in the Hudson River. And then how do these non-native species gain traction in our lake and become a problem? Well, often when a new organism is introduced into a watershed or into a lake, they lack predators from their native environment. They may lack parasites or other diseases from where they came from. So they enter into a system where they can grow freely and aggressively. And these are often what I call ecosystem engineer organisms. They are plants or animals that can reproduce early and often through multiple modes of reproduction, whether that's asexual reproduction or being able to reproduce by fragmentation, seeds, tubers, etc. And then the other parameter that we think about frequently is the water quality parameters. So we have things like pH and salinity and oxygen and light. And these organisms tend to be able to grow in a wide range of those, or have a very high tolerance for a wide range of those parameters of water quality. So they are able to establish, they start crowding out native species, mostly because they grow early, often, and more frequently. And then sometimes they can cause secondary impacts to our native species. For example, the alewife came in and is displacing some of our native smelt in the lake. And they are growing in numbers, so they're available as a forage base for our salmonids, our lake trout, and our salmon species. And when lake trout and salmon eat a diet rich in alewife, it can prohibit the transfer of vitamin B to the offspring, and it causes early mortality syndrome in those eggs. So there are primary impacts and secondary impacts of some of these aquatic invasive species. And then what are some of the actions that we can take as caring stewards of Lake Champlain to help prevent introduction and spread of invasives? Well, what you're doing today is a big one. We need early detection monitors out there looking and being able to differentiate between something that's native and possibly non-native or invasive and reporting that. There's a lot of national campaigns that we work with. Don't Let It Loose refers to pets and aquarium species that there are other options for a take back program at pet stores, or how to properly dispose of organisms so that they're not spread across the landscape. Clean, Drain, and Dry is another Stop Aquatic Hitchhikers campaign that talks about best management practices that owners of boats and other recreational equipment can take to clean off and remove mud, plants, drain live wells and bilges, and then to dry in some cases if they can't use cleaning and draining to help prevent the spread of invasive species. So there are these best management practices that we can think about following bait fish regulations. And it can expand into different realms when we go hiking, cleaning off our boots and not moving seeds around. There are different ways that we can think about how we interact with our watershed and how we can prevent the spread or the hitchhiking of organisms with us as we go.
All right. So CHAMP, we're gonna now that we know about AIS and why they're an issue on Lake Champlain, I want to talk a bit about this program as a means of addressing that issue. So this is a program run by the Lake Champlain Committee. It's a community science volunteer monitoring program, and the value of having something like that is that it helps cover areas that are missed by state personnel both in Vermont and in New York. And it just gets more eyes out on the water. Vermont has a program for invasive species monitoring on lakes, as does New York, but both of them have issues getting coverage on Lake Champlain. So it's sort of nice to combine the two programs into something that's just for Lake Champlain. It also increases awareness and literacy on AIS, and it gives really valuable data on early detection, rapid response management decisions. And as Megan noted, this is a program that is funded by the Lake Champlain Basin Program. So for CHAMP, as a CHAMP volunteer, you will do a survey in September and then a try again for another survey in early October. It's important to see how populations change over time. So if you can get more than one survey in, that's really ideal. And in this program, you'll learn about the 7 target AIS that we're surveying for out there. These are species that are already detected on Lake Champlain, as well as 9 watch list species that haven't been detected on Lake Champlain, but have been found in the Hudson River and other adjacent water bodies. And we'll also learn how to conduct and submit those CHAMP surveys. So as I mentioned, as a CHAMP volunteer, you'll do this training session as well as an in-person training session, which takes place during the week of September 15th. You should have signed up for one of those on the CHAMP sign-up form, but if you have an issue with scheduling that, feel free to reach out to me. And then, like I mentioned, conduct and submit at least one survey for AIS in September and then another in October. Try to go for 3 if you can. And in return, LCC will support you with these trainings. We'll also provide you with a toolkit, and the toolkit really is everything you need to do your CHAMP surveys. It includes a manual that contains identification resources and really detailed survey instructions. It includes survey data sheets that you'll need out with you in the field, guidance on reporting, contact information. We're also giving folks loops, which are basically little magnifying glasses you can use to help you identify smaller structures on plants and clams and mussels. We'll also be giving you an AIS survey rake, which we'll talk more about later, as well as a bin for observing species outside of the water. Also, we're giving you ongoing technical support during the field season. Really don't hesitate to reach out to me if you have questions about anything involving CHAMP. Right. So we're going to get into the 7 target aquatic invasive species that have already been detected in Lake Champlain. And these are them. It's European frogbit, water chestnut, zebra mussel, variable leaf water milfoil, brittle naiad, Eurasian water milfoil, and curly leaf pondweed. I'll pass it off to Meg for how to identify these.
Great. So the first one we're going to start with is Trappa natans, or water chestnut. Those of you who might live in the South Lake are probably more familiar with this plant than those in the Main and North Lake, although we do have a few isolated populations in Lake Champlain around Gander Bay and Black Creek Marsh, some around the Sandbar, and in one tributary on Cumberland Bay in New York. So water chestnut is a floating annual plant, and that's one of the reasons why we actually have successful management of this species on Lake Champlain, is because if we can harvest, what you're seeing here is a picture of a hand holding a rosette with little, if you see the little bubbles, they're little pockets of air that help hold the rosette up on the surface of the water. But it's a plant that's rooted into the sediments. It's native to Europe. It was confirmed in Lake Champlain in the 1940s. It came in through the Champlain Canal. It's been confirmed in all surrounding state water bodies, surrounding states that have waters infested with Eurasian water, with water chestnut. And if we can flip to the next slide. Some defining characteristics of this plant. You can see that the leaves are a triangular shape and have a serrated leaf edge. Oftentimes, if you're harvesting one of these rosettes, it's connected to 3 or 4 others, and you'll see as you pull one up, the others will come along with it. What we're trying to do is remove, in the lower right-hand corner, the green viable seeds, and the nutlets turn black when they desiccate or dry out. They're very sharp. They hurt a lot to step on, and they can cut your fingers, so you need to be a little bit careful. It does have a white flower, but the flowering period for any plants can be very short in time. So you really want to think about this one as a floating aquatic plant. Look for those giveaway nutlets, and they look like little alien heads. And if you see this plant, please remove it and do not put it back in the lake. We don't want it to spread anywhere else. But this one's pretty easy to identify. There's not a lot of other species that look like water chestnut in Lake Champlain. The next species is probably our most common, Eurasian water milfoil, or Myriophyllum spicatum. Again, another native to Eurasia. It's confirmed in many inland lakes and ponds in New York and Vermont, as well as in Lake Champlain. The defining characteristics of Eurasian water milfoil, if we can flip to the next slide, is that this is a plant that has whorls, 4 leaflets around one whorl, and the leaflets, the individual feather that comes off, usually has 12 or more pairs of leaflets on it. So we have native milfoils in Lake Champlain that often have fewer than 12 leaflet pairs on them. They also tend to look more delicate. Some of the takeaways for Eurasian water milfoil identification: the end of the leaf tip often looks like scissors have clipped it off. And then there is often a lot of internodal space. And what that means is space between those 4 leaves that come off of the whorl on the stem. There tends to be more space in between where those whorls come off the stem than other milfoil species. So this one is prevalent in Lake Champlain. I think you'd be challenged to find places that don't have it. I think you will see this most commonly. And when the plant does flower, the plant can grow in almost 20 feet of water. It'll come up to the surface, and then its bracts, or the flower stalks, will stick out of the water when it's reproducing. And another look-alike to the Eurasian water milfoil that is invasive in Lake Champlain in limited quantity, we don't have known huge dense beds of variable leaf milfoil, but is Myriophyllum heterophyllum. This one's tricky. Heterophyllum means, it's varied, right? It's going to have different numbers of leaflets coming off of the leaf itself. What it looks like when it grows in dense stands are submerged bottle brushes. Um, that's what folks often refer to it looking like. This is a perennial. Again, it's native to Europe. It's been found in Lake Champlain since 2011. I think we first detected it in the South Lake, and there was one small population up in Missisquoi Bay. I haven't seen a lot of it, and I've seen a bunch of samples coming out of Lake Champlain, but it is possible that you would encounter it in Lake Champlain. It's confirmed in other states, and we can flip to the next slide. So here you can see variable leaf milfoil, similar to Eurasian, has this emergent flower when it reaches the water surface. And it has these bracts, or what looks like more spiny leaves that come off at the tip of the plant. Again, it's feather dissected. It has whorls of leaves, and it can have 4 to 6 leaves that come off of the whorl. Again, I think the sketch, the black and white sketch on the left showing the emergent flower, showing how much more closely those whorls come off of the stem, leads to that appearance of a bottle brush underwater. Okay, next slide. And here we just threw in some native submersed lookalikes to the Eurasian water milfoil and the variable leaf milfoil. So first of all, we have native milfoils in Lake Champlain. Those could be confused with them. But we have some other species that when submerged can look like these milfoils. We have a native bladderwort or utricularia species. And if you don't take the plant out of the water and float it and look really closely at the leaf structure on the stem, it might be hard to differentiate between the milfoils. Another one is coontail. That's a common native species that we have in Lake Champlain. You will see it frequently. And the difference here is that the leaves themselves fork. They don't look like individual feathers that come off of the stem. They are forked. And again, you may need to float that plant in your little white sample tray and take a closer look to be able to see the different structure in the leaf. Next slide. European frogfoot, Hydrocarius morsus renai, is a floating leaved perennial plant. This one is very common in some areas of the lakes. It likes backwaters and wetlands. It is free floating. It's not rooted to the sediment. It's confirmed in Lake Champlain. We have it in a number of other water bodies in the watershed and in surrounding states. Next slide. So if you are able to pluck this one out of the water and it doesn't come out with like a dense rhizome or root attached to it, that's usually our native lilies have a rhizome that's connected to them. This one has a rounded leaf edge, and they can be connected to other growths on a stolon. And so multiple little groupings of the frogbit pads may appear on the surface of the water. It's very easy to pluck out of the water. This one is, again, that heart-shaped leaf. They don't get much larger than like a quarter or so in size. They can get a little bit larger, but you're looking for these mostly in quieter backwaters along the shorelines of Lake Champlain. Okay, the next species is another common one, the zebra mussel or Dreissena polymorpha. Zebra mussels we have throughout Lake Champlain. They're not doing great in Mallett's Bay, which is a good thing for those who live in and around Mallett's Bay. Zebra mussels, you will likely encounter them because they like to grow on the stems of plants, or if when you're doing your rig toss, you retrieve native mussels or a rock or something else, they might be attached to that too. Less likely that you're going to pick up individuals with the rake unless they're attached to something else. But these are small freshwater mussels, native again to the Caspian and Black Seas, likely arrived in ballast water to the Great Lakes and spread from there. The telltale difference between zebra mussels and that are present in Lake Champlain and the quagga mussel that we'll go over later is that this one is in a D shaped, and the patterns and the coloration can vary, but it has a flat side that you can often stand it up on. They're small, right? Less than 1.5 inches. And they have abyssal thread, which is looks like a series of little fine threads that come out of the mussel where it attaches, and that's very sticky in how they attach to different types of substrates. So common in Lake Champlain, found in Lake Bomazene. Let's hit the next slide. Okay, zebra mussel life cycle. We put this in here just to explain how many and how frequent these organisms can reproduce. So they create a very small veliger, which is an early life stage or juvenile form of the zebra mussel. In our long-term water quality monitoring programs on Lake Champlain, we do plankton tows and look for zebra mussel veliger densities and track that over time. And that's an early detection tool we use for other inland lakes. But the females are able to lay eggs once the temperatures rise roughly above 50 degrees. They hatch, they can float in currents and then settle and grow. And some of you may have seen them growing on top of native mussels or other native species. They're not seeking them out. They've found, what they have done is found a hard substrate upon which to grow. And so they live generally for 2 years, but they can survive for longer, and they just are prolific breeders. So we have so many of them. Next slide. Okay, switching to brittle naiad, Najas minor. So this is a different, this plant will occupy a different space in the habitat. It's a submersed annual species, native to South America and the Southeast US. It is confirmed in Champlain and a number of other inland water bodies and surrounding states. Next slide. This one grows in the sediment on the lake bottom, so it's not going to grow very high up in the water column where it's very, very dense. It can grow maybe like 2 feet or so above the bottom of the lake, but this one isn't coming to top out at the water surface like the Eurasian water milfoils. So this plant it's difficult to tell it apart from other native species. The plant leaves have minor serrations on them, and they come off the stem opposite of each other. So not an alternate is coming off one on one side of the stem and the other later on the other side of the stem, but opposite at the same point on the stem, opposite each other. It can appear whorled, but it is not whorled. You have to get out that little hand scope to take a look at it more closely to see that the seeds exist in the leaf axils. But this is one that you may encounter, and it's one that likely takes floating it in your little tray and comparing it to other species to be able to identify. We have some native submersed lookalikes. We have native naiad species, slender naiad more commonly than, Najas flexilis and Najas gracillima. I don't know if I can say that quite right. But those do exist in Lake Champlain. So this one's a hard one to detect, and it may be that you can just get it to the naiad species level, and then you need to take a sample or something to take a closer look at it. Okay, curly leaf pondweed. This one is our only Potamogeton crispus. This is the only Potamogeton of which we have many in Lake Champlain that is invasive, and it is telltale. This one luckily has these lovely lasagna-like leaves that are very easy to tell apart from other native plants. They have a serrated leaf edge if you look at it closely, but this lasagna looking like leaf structure will really tell it apart from other species that you'll see in the lake. Next slide. So we're switching over now to species that are on the watch list, meaning that they're not present in Lake Champlain now. And so we have a handful of additional plant and animal species that we want to review, so that you're thinking about these when you're pulling in the plants and making sure that we don't have these in Lake Champlain. Okay, so I feel like I need to slow down a little bit. It's a lot of information to take in at once. Don't worry, you will have guides with you and you can take pictures and you can always take samples if you're uncertain. I look at tons of samples all the time, and there are still times when I need to stop and go to a few different ID books to see what I think I've got. So fanwort, or Cobumba carolinea, this is a submersed perennial species. It's native to Southeast and South America. We do not have it in Lake Champlain. Just so you know, we intercepted it on a watercraft coming from Massachusetts this summer by a boat launch steward and prevented it from getting in the lake. But we have, there are populations surrounding this watershed. So there is a mechanism by which they could be introduced to Lake Champlain. So it's great that it's on the watch list. Keep an eye out for it. Okay, next slide. How do you identify fanwort? The way that you're going to be able to tell fanwort apart from other lookalike species is that there are opposite branching leaves. That's the first one. The second one is that there is a stem on that leaf. So the main stem of the plant runs up, and then off of it, there's a little segment of stem before you have those opposite leaves. And that little stem there is how I tell it apart from other native water marigold or other species that look very similar. So branching leaves, opposite. They do have a white flower, but again, it's rare that you're going to see the flower when you're taking your samples necessarily. Next slide. And here are some of these native lookalikes. The difference here when we have water marigold, Bidens beckii, this again has in the lower picture that very clear branching leaves, but there's no like stem on that leaf connected to the main stem. So that's how I tell it apart. And then you have water buttercup, which is ranunculus. That one's different too. That's the black and white drawing in the middle top of the screen. It looks like there's a little cupped sleeve around the leaf as it comes off of the stem, and they're opposite. They're alternate. They're not opposite. So that's how you tell another native, the ranunculus water buttercup, separate from that invasive fanwort. Next slide. Hydrilla. Hydrilla verticillata. This one scares me because it is present in states around the watershed. We do not have it in Lake Champlain. It's not yet found in Vermont. And this is a very, very aggressive aquatic plant. It looks very similar to our native waterweed or Elodea. Waterweed is Elodea species, but it is, we'll provide in the next slide how you're going to tell it apart. So hydrilla, a few things. You usually have more than 4 leaves on a whorl around the stem. The leaf edge is going to be serrated, and on our native species, it's a smooth edge to the leaf. This species has had tubers, or it's that little white-shaped growth that grows over winter in the sediments. So it's not going to pop off of the side of the plant necessarily, but if you were to harvest this plant and get it out of the sediment, you might be able to pull up that winter growth, the tuber that overwinters in the sediment. This plant has turions, so it can spread by seed, but it is a very aggressive growth. This plant can grow and is expected to outcompete Eurasian water milfoil in water bodies if it gets in. Next slide. Another invasive aquatic plant that looks like hydrilla is Brazilian Elodea, or Aguirrea densa. This is a much more robust plant. It's again a submersed perennial species native to South America, not found in Lake Champlain or in Vermont. It is, I think, in a few places in New York. Next slide. Brazilian Elodea, again, has these lance shaped leaves, whorls of 3 to 6. I think I have one more slide to talk about Brazilian Elodea here. Oh, it's a native submersible. Okay. So basically, if it's native, it has 3 on the whorl and it's a smooth leaf. If it has more than 3 leaflets on the whorl, then you want to report this, take a picture, or take a sample. It could be a Gyriadensa, it could be hydrilla if it has a serrated leaf edge. But again, the Gyriadensa or the Brazilian waterweed would have more than 3 leaves on the whorl. And then the hydrilla would have more than 3 leaves on the whorl plus the serrated leaf edge and possibly have the tuber growth. But we have tons of Elodea canadensis, our native waterweed or our slender waterweeds, the Nutali, in Lake Champlain. And again, I think you will see it nearly as common as the Eurasian water milfoil or the curly leaf pondweed or maybe native coontail. There's a lot of Elodea in Lake Champlain. Okay, parrot feather is another myriophyllum. It's an aquaticum. It is a little bit more delicate. It does look like a parrot feather to me. It's a rooted submersed plant, again, native to South America, not found in Lake Champlain, confirmed in surrounding states. Next slide. Some of the defining characteristics of parrot feather, again, we're going to have whorls of greater than 4, so 4 to 6 leaves. And the leaves themselves have 10 leaflet pairs. The leaves can extend above the water surface. So this plant will grow underwater, and then once it gets to the water surface, it will come out of the water a bit. It's got more structure to it. It's a bit more brittle. And if the plant is flowering, this is an unusual but a helpful identification characteristic, is that it has small white flowers that grow in the axis of the leaf and the stem. So there's a defining characteristic for you. It's okay. Go ahead. You can switch this slide. Yep. Oh, that is not starry stonewort. Maybe this is an extra slide. Oops. Okay. It's okay. Go back. Starry stonewort, that's parrot feather. Somehow I put the wrong photograph here. We'll fix this. This is supposed to be a bulbil photograph. So starry stonewort is different. Starry stonewort is actually a macroalgae algae species native to Eurasia. It has been established in Lake Derby and Lake Memphremagog, not known to occur in Lake Champlain. It is confirmed in New York and some Midwestern states. Next slide, we'll show you what it looks like.
One thing, if I just scroll back to the intro slide for the
That's the
watchlist
bulbil.
species, right here on the bottom left,
Thank you.
that's what you're looking for on starry stonewort, is that little, what is it called again, Meg?
It's called a bulbil, and that was going to be the leading photograph. And I apologize that there's a parrot feather photo where there's supposed to be starry stonewort, but that's a bulbil, and it's a growth that occurs on this macroalgae that has the form. It looks like it's an underwater aquatic plant, but it's actually an algae. So let's go back to the second starry stonewort slide, and we'll talk about it a little bit. Perfect. Okay. So this one's confusing. Let's see. So it's an algae. Again, this algae grows, and then there at the node, it's going to separate and grow. 4 to 5 different branchlets will come off at one node. There'll be different lengths. There'll be different branching variation. And in order for it to anchor to the sediment, it uses like this clean filamentous, it almost looks like fishing line growth to stay sort of connected to the bottom sediment. So this one is not going to come out of the water at the water surface. This is going to grow only at the interface of the sediment bottom of the lake and remain a low growing algae. So at the nodes of these different segments of growth along, we're going to call it a stem because that's easier to communicate. Sometimes these bulbils grow. So those white little star-shaped bulbils. And so far, only males have been found in North America, and reproduction can occur from both the bulbils and from fragments of the algae. So this is a tricky one to identify. I think it can look like a lot of our native naja species or naiad species in the lake. The difference here is this one grows outward, so it's wider in its branching growth, and that it is got a lot less. It's not as compact around the stem. Let's just put it that way. So where it grows, it can grow, and when a bunch of these algae grow next to each other, it can just look like a bed of low growing like grass on the lake bottom. Next slide. Okay, and then we're going to flip over to water soldier. This one, again, I would be shocked if we encounter in Lake Champlain, but it is good to look for. We've had no reports in Vermont in Lake Champlain, but there have been reports of this species growing up in Quebec and Ontario. So it can somehow make it up this far and grow and through our cold seasons. So this is a loosely rooted perennial submersed and emergent plant. It's submersed and emergent because it starts growing out of the sediment underwater, and then when the water temperature warms up, it floats to the top and it grows. It looks a lot like an aloe plant. It's native to the UK, Spain, Italy, Bulgaria, and Siberia. Next slide. And the water soldier it's buoyant. Its leaves are shaped like, again, serrated swords with sharp spines. And it has white flowers. Again, not a great telltale because it may not be flowering when you see it. If you are going to see this plant, it's likely once the water temperatures have warmed up, it floats to the surface and then it becomes emergent at the water surface. So a very sharp, it will cut your hands. It's very rigid plant that grows at the water surface. Next slide. Okay. Corbicula fluminea. This clam is unique. It looks different from all of our native clams because it has these raised concentric ridges, and the coloration can change. I've seen them purple, white. I've seen them yellow, brown. They tend not to get larger than a quarter in size, but if you take an individual clam and take your fingernail and run it over the surface, it's going to click. You're going to hear that. Those raised concentric ridges are unique to this invasive, and all of our native species in the lake do not have raised concentric ridges on their shells. So where you would encounter this species would be primarily if you're doing a sediment sieve or your rake goes into the sediment, maybe you'd pull one of them up. We have not documented them in Lake Champlain. They are hermaphroditic, which means it only takes one of them. An individual can reproduce. It has male and female parts and can create and release juveniles into the water. They usually float with the current, settle in the sediments, develop into these clams, and the clams have a pedal foot so they can move in limited distances around in the sediment and filter feed. So we have an infestation of these in Lake George. We know they're present in the Champlain Canal and in Lake Bomazene, but we have not documented them yet in Lake Champlain. Next slide. So in the areas where we have seen them colonize, they are able to rapidly reproduce, dominate the habitat, push out some native species. And we've had some aggressive management in Lake George that was largely successful with adult clams, but the juveniles are very hard to both detect and to manage in any effective way. we were able to manage the adults by using benthic barrier mats to cover them up and smother them. And that worked very well, but the juvenile life stage was not possible to manage. So they are definitely present in Lake Champlain, and that photograph right there is a fond one because that's from our partners at the Lake George Association. Round goby, Neogobius melanostromus. I spend a lot of my time on round goby. This is an invasive benthic fish, meaning it tends to hang out at the substrate or the bottom of the lake. It likes rocky areas with a little bit of current. Native to Europe, first found in Lake St. Clair in 1990, spread throughout the Great Lakes within about 10 years or so. Moved across and through the Erie Canal to the confluence of the Mohawk and the Hudson River, and are poised to try and start coming up the Hudson through the Champlain Canal into Lake Champlain. There's another population in the St. Lawrence River, and that population equally can threaten to come up the Richelieu River towards Lake Champlain. So we have a very active early detection monitoring program for this species. It looks a lot like our native sculpin. And the difference here, which is critical, is that round goby has this fused pelvic fin that looks like a suction cup. And that fused pelvic fin is the defining characteristic to tell it apart from the slimy sculpin, which has the splayed pelvic fin underneath on its belly. So they don't get huge, 4 to 10 inches in length. They are probably expected to do well in Lake Champlain if they get here. And we have had some reports from anglers of reporting suspicious goby-like looking fish. And luckily, to date, all of those investigations have turned out to be the slimy sculpin and not the round goby yet. Next slide. Why we're worried about it is that it likely will outcompete our native benthic species like darters and sculpin. They are very aggressive egg eaters, and we're concerned about them eating eggs of bass and lake trout and sturgeon species. They are able to spawn multiple times a year again. And in ecosystems or in lakes where they have reproduced and started to cause impacts, it's also changed the angler experience because they're such aggressive bait fish eaters. They will be the first fish to bite on a hook, which can become very frustrating if you're trying to fish for other organisms. They will eat zebra mussels. They are never going to eat zebra mussels in quantity enough to impact the population that's present in Lake Champlain. They have a pretty wide distribution, and another piece that we're monitoring is, in addition to our concerns about impacts to biodiversity and changing maybe the angling experience, they are known to carry viral hemorrhagic septicemia, which is an invasive fish pathogen that can affect over 25 different species of native fish. And that viral hemorrhagic septicemia is the whole reason why the states of New York and Vermont issued emergency bait fish regulations in the late 2000s. So we're concerned about that, and we are testing populations that are getting closer to Lake Champlain to ensure they're not carrying that harmful virus. Great. I think I'm going to switch back to Eileen here.
Yeah, thank you, Meg. I hope you're all taking that in. Just so you know, all that identification information is available in the CHAMP manual. So, you know, you don't have to memorize it all now. This is just getting you familiar with these plants, and you can always refer back to the materials that we give you in the CHAMP toolkit. Speaking of materials, the required materials for a CHAMP survey. Basically, this part, I'm going to walk you through doing a CHAMP survey. And I'm starting out with what you're going to bring with you. So what's required? Here I have everything that we provide for you in blue. So we give you the rake, which looks like what's pictured there. It's just 2 rake heads tied together and attached to a length of 25-foot rope. We also give you survey data sheets, and we give you a jeweler's loop, which is a magnifying glass to look at smaller structures like the serrations that Meg was talking about. And we also give you a white bin that is used for viewing specimens in a contained area. And we recommend that if you take pictures of plants to send to us for identification help, take the picture in the white bin, because that helps us view identifying structures on the plant. Some optional materials are, and I'll talk more about this part next, but you can either survey from a watercraft or you can survey from shore. So if you're going to survey from boat, it is required that you have personal flotation device with you, but it is optional to do that. So, or it's optional to survey from a boat. So that is all technically optional materials. You can also bring a cooler and a Ziploc bag for collecting specimens, polarized sunglasses, other sun protection measures, highly recommended, and a GPS unit, because part of this program, we ask you to take GPS points throughout your survey, but you can use the navigation app on your cell phone. Every smartphone that I know of has this feature on it, and if you have questions about accessing that, you can let me know. All right, so getting started on your survey, you want to know really where your site is and how to describe that in the survey. So we're looking at a quarter mile or less of shoreline. The start point is where you do your first rake toss, and that is the one end of your survey area. And then the end point is your final rake toss, and that's the final end. So you want to spread out the other 8 rake tosses in between that start point and end point. Before you start your survey, you want to get that start point and write down the time at which you start. If you're doing your surveys from shore, you want to space out these rake tosses linearly along the shoreline, or if you're doing it from a dock, that line's going perpendicular into the water, but same idea, you space it out evenly throughout that area. And if you're doing it from a boat, instead of being on a line, you want to be interspersed throughout the littoral zone. And the littoral zone is the area of water in a lake that plants grow. So it's that sort of shallow area where you can still see plants growing down to the bottom. Okay, so the first page of your survey data sheet looks like this on the right. To fill it out, you just sort of fill in all this information here. Let me see if there's anything I want to make sure stands out to you. Yep, like I said, before you do your first rake toss, get the time and get the start point. And if you have questions about filling in any of this part, describing your site, things like that, please either ask at the end or reach out to me. I'll make this part quick because I'll go over this in more detail for the in-person training. But if you're doing a shoreline survey, you're doing the rake tosses from shore, you want to hold the end of your rope in your non-dominant hand and throw the rake in your dominant hand. And then you let the rake sink to the bottom and pull it in. And that will help you get those submerged species that are hanging out at the bottom of the lake. You pull in your sample, and then we provide you with a white bin to examine what you gather in that rake toss. Now, a lot of times a rake toss will get you a massive clump of plants. It's recommended that you sort of pick through that clump and view it in the bin as you go through the different species. And it's really important for this part to be thorough because, as Meg was mentioning, a lot of those clams and mussels, you're not going to get a loose one. You'll get them attached to a plant, and they can be really small, especially zebra mussels. So really do a detailed job of picking through those samples. And if you're doing your rake tosses on the water, same thing, except for something I do want to know is A, PFD is required. B, something that we heard a lot last year in the program is when you do a rake toss in something like a canoe or a kayak, you'll throw the rake out, and then as you pull it, you're actually pulling yourself towards the rake because it's acting like an anchor. I just want to note here that that is okay. As long as you get the sample, it's okay that it doesn't comb across the entire lake bed. You should still get a viable sample from doing that. And once again, thoroughness is very important as you go through and identify the species. So, okay, at the starting point, record the GPS point and the time, do the first rake toss. You'll record your findings on the survey data sheet. This is what I have here is part of page 2 of the survey data sheet. After that basic info, you basically have a checklist of the target species and then another checklist for the watch list species. So if you find the species, check it off. And then at the end, you'll want to go through and categorize the area covered by that species that you've found, and also the distribution of it. For distribution, that refers to what you find on your rake toss. So the trace parameter is if you see it really just once or twice on a rake toss. Sparse is 3 to 6. Moderate is most of the rake, and dense is a large mass over the entire rake. So you do your first rake toss, record all that information, then you do another 8 rake tosses spread out as evenly as possible over the survey area and record your findings cumulatively on the survey table. Something important is for species like round goby, obviously you won't be able to survey for that when you're doing rake tosses. So you want to make sure as you're doing the rake tosses, you're also getting a good look at the water itself. Even then, it'll be hard to see something like round goby, but it's still important to know what it looks like. As you go along, record your species, and then estimate the distribution and area covered after the survey. 10th and final rake toss will be done at the end point of your survey area, and you will record the GPS point and the end time. So for suspicious species, say you come across something that you think it's an aquatic invasive species, but you're not quite sure how to identify it. Maybe it looks something like what's on the watch list, but you're not quite sure about it. For one thing, you definitely want to take photos of it. And as I mentioned, take the photos in those white bins, and then you can send those on to me and I'll share it out with Meg if I need help identifying it. And this is on the third page, this section of the data sheet. So if you find something suspicious, you record it here, and then if you think that if the photo is such that it would be difficult to identify, or if you're really feeling like it's like an aquatic invasive species that isn't, or that it's something on the watch list, we ask you to take a sample of it. So take a large piece of that plant, not just a fragment, take as much as you can so it's something we can actually identify, and store it in a plastic Ziploc bag. And keep it somewhere cool so you can put it in the refrigerator. And in the CHAMP manual that I'll be sending out, there's two different sets of protocols for Vermont and for New York on how to go about sending in a sample to the state. And that includes like mailing labels and everything that you'll need to send in your specimen. Okay, so once you complete your survey, you'll have your data sheet filled in. You want to transcribe that data sheet onto our online form, and there's going to be links to that form in my outreach that I'll do to you soon, and it's also going to be in the CHAMP manual that I'll share out. And the form is basically just a replica of your data sheets. Just transcribe it exactly as you have it written. Check every section for accuracy before you submit, and then at the bottom of the page, you'll hit this Submit CHAMP Survey button. And then once you hit that button on the bottom, it's going to prompt you with, are these values correct, with a copy of your survey. If you see something that you need to go back and fix, you hit Previous on the bottom of this page. And if you want to confirm your submittal, hit Next, and then it should go through. You'll know that it goes through because you should get a confirmation email after sending it in. And if you have any issues with sending in your surveys, of course, reach out to me. Okay, so next steps. I will aim for today, if not tomorrow, to send out a digital copy of the manual along with a link to this recording of this presentation, as well as information about getting started at your site. Um, and then next week, we're hosting our in-person trainings. You can see those training dates here. And you should have filled in one of these on the sign-up form, or you should have signed up for one of these on the registration form. We want you to choose a site and start surveying at that site really as soon as possible, because as it gets cooler, a lot of these plants will start dying back. So if we can start surveying after both training sessions are completed, the sooner the better. And if you need help determining your site, or if you have questions about your site, reach out to me. Thank you so much for watching this presentation and for participating in CHAMP's 2024 season. Please reach out to me at eileenf@lakechamplaincommittee.org with any questions you have pertaining to the program. And I really look forward to working with you all. Thank you.